It's a tremendous pleasure to be here today to talk about California dreaming. It was so nice to have Mark and Jeff lead off the session. They've been tremendous partners to us in our research. Not only are they phenomenal scientists, but they're practitioners. They really, put science into action and bring the practicalities of growing tea, bring it into the real context. But I hope that today you'll join me in a little journey of California dreaming. I, like Jeff, grew up here in California. I actually grew up in Sacramento, and I'm convinced that one of the earliest events in my lifetime, the lunar landing, planted a seed in my psyche that has continued to grow. It gave me visions of vast possibilities. I could have never dreamt then that one day I would have the opportunity to actually work at a university in the Department of Chemistry that actually trained an astronaut. Tracy Caldwell Dyson is my hero. She made her dream come true of becoming an astronaut, a space shuttle astronaut, after earning her PhD in chemistry here at UC Davis. Tracy always reminds me to dream big, but I don't think that when we met, she could, or I could have imagined that one day my biggest dream would be to be a farmer. This was a tremendous opportunity for me. This just took place this last year. I had the opportunity, because of the GTI and Catherine's leadership, to go to India, to the Toklai region of India, and plant our first joint plant with UC Davis and the Tea Research Association in India in a research field in India. It was a bit of a journey to that point, and I'd like to tell you about that now. A little bit about how an aspiring astronaut ended up with the biggest dream being a dream farmer. And the answer actually to that question of how it happened is chemistry, collaborations, and microbes. So hopefully you'll be a little intrigued by that. My talk today, the story I'm going to tell you about is growing tea in California. You've heard a lot about that already. Reality and vision. And so this is a map of the major tea growing regions of the world. You'll notice that California is not on this map. Of course, China is one of the largest producers, and this is the area of Assam where that previous picture where I was up in India. So I was up in the very northern part, right near the China border. But what I want to tell you about is how this story has unfolded for me as a result of my ability to go to Taiwan. I've been going there for over a decade now and really enjoyed important collaborations during that time period. And so I want to tell you kind of in chronology how this all came about and how we came up with the idea that really couldn't have been appreciated back in the 1960s when we began growing tea here in California, and that is the importance of microbes. That's really a 21st century idea. Our pathway into the chemistry of microbes, if you will, actually began with a human condition, H. pylori. H. pylori, some of you may have heard about it. It's a bacterium that infects half the world's population. Most people don't even know that they have it, and in fact, it's believed that it can be a very beneficial symbiotic relationship. This bacterium appears to have co-evolved with humans over a very long period of time. It can transform. It can undergo a transformation that can lead to cancer progression. And my collaborators in Taiwan contacted us because of our chemical skills. We're actually able to take compounds that can be consumed by microbes, and we can modify those compounds. We can put a tag on them, and different kinds of tag. It could be a stable isotope, it could be a radioisotope, it could be a fluorescent molecule that could cause the bacterium to light up. Because of our chemical skills, we can make molecules like this. We can feed them to microbes, or in this case, H. pylori, and then we can see what kinds of molecules that bacterium is capable of making. When we did that with H. pylori, we found that it made rather complex molecules. But in order for the H. pylori to be virulent, to actually lead to the progression of cancer, it had to make a specific compound where it combined molecules that the host gave it as well as its own into a structure that allowed it to gain access to the host cells. So in this way, the bacteria modulates the membrane of the host cell and can then inject its virulence factor into the host cell. And then that is what leads to cancer progression. We're continuing these studies. We're not quite sure what causes the bacterium to gain virulence in the first place, and understanding that chemistry is very important to us. But every milestone along the way, we make sure we celebrate. So when I was in Taiwan doing these studies, my colleague, Hans Lin, invited me to my first tea plantation. He took me up to Alishan, which is the high mountain tea region of Taiwan, and immediately I was struck by the similarity to the wine country here in Napa Valley. So as Jeff had just pointed out, how we asked, why don't we grow blueberries in California? I asked Hans, hey Hans, why don't we grow tea in California? And Hans laughed. And he said, it's because of the water, the soil, the weather. And he explained to me this concept of all environmental factors that contribute to the phenotype of the plant. And California doesn't have any of them. And what he said is it's really linked to a specific habitat. And I said, yeah, Hans, but what if we could change the microbes to change those factors? He's like, that's a pretty interesting idea. He then explained to me about tea. I knew nothing at the time, absolutely nothing about it. And he explained to me that true tea comes from the plant Camellia sinensis, and that there are two main varieties of this tea, sinensis, meaning from China, or Assamica, meaning from India, the Assam region that I pointed out earlier. And from one plant, one can make all of these types of tea, as Mark and Jeff have pointed out. And so at this juncture, I really became intrigued by the idea that perhaps we could use microbes to influence the ability to grow tea. I mean, I knew nothing about growing plants at all when we began this, and so I first started just looking at tea commerce and trying to understand the history of how tea has developed over thousands of years. But I started with more recent history, maybe the last 200 years, and what I learned, starting from now going backwards, something really caught my eye. Taiwan, for such a small country, actually has a large growing region. Japan's about twice that much. Of course, China is the #1 grower of tea, and India is close behind. And what I learned was that the modern tea industry in India actually started in the 1860s. I was surprised to learn that. I thought it must have started much earlier than that. And documents indicate that it actually took about many, many years to get that established, maybe about 60 years. And I thought to myself, during that time, the weather didn't change very much. The water didn't change very much. But maybe the soil did. Maybe microbes in the soil changed over that period of time that led to India becoming the second largest producer in the world today. And so this then, actually those studies brought me to a very interesting finding. It turns out that at the same time that tea was beginning in India, it was beginning right here in the United States. It was beginning in South Carolina. The USDA appointed Charles Shepard, who I might add was a chemist, to lead the project. And the USDA brought in seeds from China, from India, Japan. In their documents, they refer to it as Formosa, which of course is Taiwan. And Dr. Shepard grew about 50 acres of tea in South Carolina. He, over more than a decade, he looked at producing the seed, growing the different cultivars, the soil, water conditions. He determined that it could be done. One of the most successful teas was Dragon's Pool, and this actually came from, in the documents, it's read as Hangzhou, which is today, we spell it a little bit differently, but Hangzhou in Zhejiang province in India, and in China. And it turns out that actually the tea was very good. He won the World's Fair for oolong tea in 1904. And there's going to be a little link here to the oolong tea that Jeff was talking about here out at Kearney. So going forward from here, one thing that Dr. Shepard noted, which we have said time and again here, was labor. Labor, even back then, was a problem. And here is a picture of the plantation from the late 1900s. And you'll notice that the farm laborers are African Americans, mostly children and women. And from what I can gather from the documents, the laborers actually lived on the farm and were educated there as well. So that made it worth their while. But you can see that it was growing quite well. So fast forward to the 21st century, where actually I believe this is a miracle. So we're really actually in Taiwan, and I see coming across my screen that the Global Tea Initiative is being started at UC Davis. And at this time, I had been studying tea for about 3 years. Not doing research in it yet, just understanding the context of what was needed in terms of research. And I was just like blown away. So this was 3 years ago. And shortly thereafter, when I returned, we had the first symposium. And I got to meet Catherine there. And because of Catherine's leadership and the strategic partnerships she's put in place, we have been able to achieve a great deal in the last couple of years. So a debt of gratitude is owed to you, Catherine. Thank you very much. So what Catherine put Jeff and I together. Jeff got that phone call because Catherine said, I was out at Kearney and I saw tea plants. And I was like, are you kidding me? And that's one thing about Catherine. She travels all over the place. I imagine most of the people in this room are here because they met Catherine out traveling on behalf of the GTI and promoting the GTI. So the documents that Jeff deposited from Kearney indicated that actually Emil MARAC was very instrumental in this first study. He was a food technologist. He was actually our first chancellor, and MARAC Hall, just up the road here, is named after him. And as Jeff has told you, it was Lipton that was interested in doing the studies. And Carl Ingebrigtsen was one of the lead scientists here at UC Davis. And as Jeff has indicated, Carl showed that he could grow high-quality tea. He applied water as overhead sprinklers, as Jeff noted, and they surmised that might have actually increased the humidity. And for that reason, weather was really not a problem out at Kearney. And they planned to do cutting by automation, and they actually did in 1978. So this is a picture of the field that was growing out there at that time. I don't have any color pictures that I could find of that time, But this is a modern tea field in Taiwan with one of our collaborators that we work with. And this is what the overhead sprinkler system looks like. So this is being used today in Taiwan, this overhead sprinkling approach, even though they have a lot of rain. Now, this actually is this is a current plantation in South Carolina. It's called the Charleston Tea Plantation. This is what they call the Jolly Green Giant, and this is probably the automated cutter that they used at Kearney back in the 1960s. The researchers that started this tea plantation were part of the Lipton Project, and they had been at Kearney working back in the 60s. This is a rather, they're still using this today. It does automate cutting of the fields, and you can see that the fields are engineered for it, but it's not very precise. It cuts about 6 inches off the top of those fields. This is a more modern type of an approach that's occurring now in Taiwan, where the fields are these are actually double rows of plants, and this is a Japanese precision cutting machine. And you can see that you can actually get much finer cleavage of the top of the plants. So I think, as everybody's noted, this is still a challenge. I think that there can be innovations made in this area. But one of the things that we're interested in is moving this from the current reality into a vision of really having a sustainable tea growing enterprise in California. So as chemists, our first approach to anything is to look to the literature, see what others have done, and try to repeat it. So that's exactly what we did. Because we had these rich experimental documents from Carl Ingbertsen's lab, we tried to reproduce those. These studies started less than a year ago. And lo and behold, we were able to reproduce their studies. This is Crystal Yi. She's one of the leading graduate students on the project, holding one of our plants that we started propagating, like I said, less than a year ago. So currently we have, from the 14 mother plants out at Kearney, we have several hundred plants that are ready to go into the ground as soon as Jeff gets that plot prepared for us out there. So this is the reality. I think we can all agree we can grow tea in California. It's no question important economic considerations exist. And as chemists, we're asking the question, what can we do to promote the growth of tea in California? And what I want to tell you about now is a platform that we imagine where we increase the germplasm that's been talked about. So the scientific platform, that would depend upon genealogy and genetics, and then also promote the growth and production through chemical innovations and encouraging appropriate microbiomes for the plant. So our studies actually were really facilitated by, last summer, the first release of the genome of tea. This was actually a huge endeavor that enabled us to work with the USDA to trace back the plants that are growing at Kearney to their genetic heritage. And this is done with Jeff, and again with the researchers at the USDA in Beltsville, Maryland. And what we were able to determine is that we do indeed have, at first we didn't know of the 14 plants out there. Are they all the same? Are they different? Turns out 12 of them are one cultivar, 2 of them are another cultivar. And they trace back to a Chinese clone that is currently growing in Assam. So what we believe is that, and we know that these plants came from South Carolina when the study was begun. So we believe that these are the plants that Charles Shepard was working with on his plantation. Now, we've been able to go further and take the next step. So the next step is, now that we know the genetics, we know what the plant can do, what about microbes? Are there symbiotic microbes associated with these plants? So in collaboration with Johan Laveau here at UC Davis in plant pathology, we were able to go out and collect roots. A root specialist here on campus, Astrid Volder, joined us, thank goodness. She showed us what a root looks like, the appropriate route, at least, for getting symbiotic microbes. And we were able to bring those back to Johan, and he sequenced them. And he showed that there are definitely symbiotic microbes associated with the plants, and they're cultivar specific. So the plants clustered into one type of microbial association versus another based upon their genotype. And this is exactly what we were hoping to find, because we know that that's exactly what happens with H. pylori, that the microbe, the different genotypes have different chemical possibilities. So going to the next step, we were now ready to begin to look at the chemistry of these interactions. And so this here is our plants growing here, just another plant. You can see they're quite vibrant with a very nice root system. This was grown in a greenhouse. But just to pictorially visualize this, what we have is like the root of the plant here. And inside the root, these are buscular mycorrhizae, for example. That's what's shown here. These symbiotic bacteria, their membranes have to coexist with the membrane of the plant. And what they do is they cross nutrients across that membrane. So for example, the green part of the plant will capture the sunlight and produce carbon-type molecules, like carbohydrates, for example, and feed those to these arbuscular mycorrhizae. They will then uptake these nutrients and then feed them back to the plant. And it's that chemistry that we're equipped to now study because of the fact that we can put these probes on all of those molecules and follow them in the different processes. So we're set with this platform to go forward. And I just want to tell you about one important process that's important in the sustainability of any plant, and certainly pertains to tea. And that is phosphorus cycling. So as many of you might realize, we often will apply phosphorus as a fertilizer or as a nutrient to the soil. And too much fertilizer can end up in the streams and be an environmental hazard. So really being able to control phosphorus cycling is important. So one thing that happens in plants is if all of a sudden they don't get enough phosphorus, they start breaking down their phosphorus components. So they might take a phosphoglycerolipid, such as this, and degrade it into phosphate and a glycerolipid. That then can get attached to a carbohydrate by an enzyme to form a structure that might look like this. These compounds, these are glycolipids. They have a carbohydrate and a lipid component, and they can replace up to 70% of the plant membrane. If there's now a microbe associated with that plant membrane, these compounds can be passed to that microbe. The microbe can break them down into various different components and then reassemble them to give it back to the plant. And in this process, the microbe is able to take phosphate from the environment and re-give it back to the plant. So this is one important process that we're following in our laboratory to help facilitate the growth and production of tea in California. We can really get a chemical view of this, not only because we can make our labeled food, if you will, we can feed these components to a plant, but because of another innovation that was made possible by a graduate student in my research group, Amy Bellingeri, and collaborations with David Tricoli in the Plant Transformation Lab here at UC Davis. We're actually able to grow tea not in soil, but in a petri dish, if you will. This is a microbe-free environment. We can completely define every ingredient we put into this medium, and the plant grows on this. This is tissue culture growing of tea. And because we have this, we can now feed our microbes feed our probes to this plant and see the kinds of compounds that it's able to produce. Now, this is in the absence of microbes. Because of Jeff's work and the wisdom of the people to keep the plants that we have growing out at Kearney, we're able to to actually also apply our compounds to the leaves of these plants, for example, to the roots of these plants, and study the chemical compositions that might come out of those. And so in this manner, by comparing these two outcomes, we can see the role that microbes are playing. And because we can profile the microbes and know their identity, that gives us the opportunity going forward to create what we call symbiotic communities that we can use to promote the growth of these plants, and in particular, the growth of tea in California. So with this platform in place, one of the biggest visions that we have going forward is that we can actually combine our work with human microbes and plant microbes in understanding this global exchange of these chemical components. We know that roots of the plants, of our tea plants, for example, have microbes associated with them that are producing compounds that are very similar to the microbes that are in the human microbiome. The question that we're asking is, does the plant make do microbes associated with the roots make compounds that then go out onto the tea leaf? Humans consume them. They may feed our own microbiome and contribute to our health effects. It's a big journey that we're embarking upon. We've only just begun. It wouldn't have been possible if we didn't have the plants out at Kearney. It wouldn't have been possible, and that we wouldn't even know about if it wasn't for the GTI. So this has just been a remarkable journey for us, and we're looking forward to continuing it. This is my current research group. These are the chemistry pioneers. You've seen a lot of the other pioneers already earlier. So this is Crystal. She's going to actually be talking tomorrow afternoon about her chemistry that she's doing. Amy Bellingary, who's doing the tissue culture studies. Amy and Shen Yang have just joined the group, and they're continuing on. Amy is very interested in what ends up in the cup, and Shen Yang is very interested in this membrane-membrane interaction. I have two phenomenal undergraduates working with me, Dewey Pham. Dewey is in charge of working out at the greenhouse with Bill Werner, who has been just a tremendous resource for us. We wouldn't have any plants if it wasn't for Bill. And Danielle's really interested in the phosphorus cycling. Now, UC Davis is just a tremendous resource for us, and I've already mentioned all my collaborators here. It's Hans Lin at Academia Sinica started all this with our H. pylori studies, and Chi Horn Kuo is there in their agricultural research station helping us doing microbiome studies in Taiwan. So we really want to compare the microbial communities in California versus those in Taiwan. Finally, I would like to say that I hope, I think with all these resources in place and with this great audience here and the support that we've enjoyed, that we can do it. I think we can really grow tea in California and make it a viable crop. And I don't think it's really that far-fetched. I think that there are bigger dreams that are even coming true today. So all it takes is the 21st century innovators. And I, like Jeff, am really looking forward to seeing the young people come up and lead the way. So thank you very much for your attention.